Imagine a world where diseases like cystic fibrosis, sickle cell anemia, or even certain types of cancer could be erased from our DNA, like fixing a typo in a book. But if we could rewrite the code of life, should we? The power to alter DNA comes with profound ethical questions that scientists and ethicists are only beginning to explore. It might sound like science fiction, but this is the reality of gene editing with CRISPR, a revolutionary tool that allows scientists to rewrite the genetic code of living organisms with incredible precision. But how does it actually work? At the heart of CRISPR is a natural system that bacteria have been using for millions of years to protect themselves from viruses. Bacteria store fragments of viral DNA in their own genetic code as a kind of memory bank. If the same virus tries to attack again, the bacteria use these stored sequences to recognize the invader and cut it apart before it can cause harm. Scientists realized that they could harness this system to edit DNA in virtually any organism, including humans. The CRISPR system relies on two key components, a guide RNA and a protein called Cas9. The guide RNA is a custom-built molecule designed to match a specific sequence of DNA. Think of it like a GPS that directs the system to the exact spot in the genome where a change needs to be made. The Cas9 protein acts as molecular scissors, cutting the DNA at that precise location. Once the DNA is cut, the cell's natural repair mechanisms take over. Scientists can then tweak these repair processes to add, delete, or modify genetic information. One way scientists use CRISPR is to disable a faulty gene. If a gene is responsible for causing a disease, cutting it can stop the harmful effects. Another method involves replacing a defective gene with a healthy one. Scientists insert a new DNA sequence at the cut site and the cell incorporates it into its genome. This approach could potentially cure genetic disorders before they even develop in a person's body. CRISPR is already showing promise in medical treatments. In clinical trials, researchers have used it to treat blood disorders like sickle cell anemia, where faulty genes cause red blood cells to take on an abnormal shape. By editing the patient's DNA, scientists can correct the problem at its source. In the future, CRISPR might be used to treat more complex conditions, including heart disease, and even some forms of blindness. Beyond medicine, CRISPR is transforming agriculture. For example, scientists have used CRISPR to create disease-resistant rice and tomatoes with longer shelf lives, reducing food waste and increasing global food security. Scientists are using it to create crops that grow faster, resist pests, and survive harsh environmental conditions. This could help address global food shortages and reduce the need for harmful pesticides. In the field of conservation, CRISPR could even help revive endangered species by boosting genetic diversity. Despite its incredible potential, gene editing with CRISPR raises ethical concerns. In 2018, a Chinese scientist announced he had used CRISPR to edit human embryos, sparking a global outcry and leading to debates about the risks of altering genes that could be passed down to future generations. Should we edit human DNA to enhance intelligence or physical abilities? Could altering genes have unintended side effects that we don't yet understand? These are questions that scientists and ethicists are debating as CRISPR technology advances. Laws and guidelines are being developed to ensure that gene editing is used responsibly and safely. CRISPR has unlocked a new era in genetic science, offering possibilities that were once unimaginable. While there is still much to learn, the ability to edit DNA with such precision is already changing medicine, agriculture, and our understanding of life itself. The future of CRISPR is still being written, and as technology continues to evolve, so too will the ways we shape the very code of life.
Got a question about how something really works? Drop it in the comments and we might cover your topic in our next video.